So welcome everyone to a very special edition of our Global Health Rounds. We are truly honored today to have our speaker, Dr. Michael Houghton. As most of you know, he is the director of the Li Ka-shing Applied Virology Institute and professor in the department here, U of A, Department of Medical Microbiology and Immunology. He's an international leader in viral hepatitis, and as I think we all know, the most recent Nobel Prize winner 2020 for physiology and medicine for his discovery of the hepatitis C vaccine. So we are very happy to have him here. I'm going to keep this short. I could probably keep going through the introduction for the next half hour, but Dr. Houghton has done amazing work, him and his team. He's received numerous other international awards prior to the Nobel Prize as well, and his passion is the application of biomedical research to prevent disease. And today he's going to be talking to us on towards the control of hepatitis C, COVID-19, and whatever is next. So Dr. Houghton, I will leave it to you. Thank you.
Thank you, Dominic, and thanks very much for the invitation to address the Global Health Group. It's my pleasure. So I'm going to talk about a few things, as Dominic said. I'd like to talk about Hep C to begin with. So right now, hepatitis C is itself a pandemic. WHO estimates there are 2 to 3 million new infections occurring globally every year. And around 400,000 people are dying from hepatitis C associated disease. In the US, CDC estimates there's around 20 to 30,000 new infections every year of Hep C. Most of those occur in people who inject drugs, intravenous drug users, and with the growing epidemic of opiate use, fentanyl and so forth, I believe the transmission is actually increasing. The incidence of infection is increasing in the US and in Canada. In Canada, the PHA, Public Health Agency, estimates there's up to 12,000 new infections every year in the country. And once again, as in the US, most of these, around 70%, occur in people who inject drugs. And that's an easily forgotten group of people, unfortunately. Canada does a very good job trying to reduce harm reduction in people who inject drugs. And a lot of people, I think, forget that there's a reason why people who inject drugs, most of those people have had a trauma, some kind of very severe trauma in their lives, often in their young lives, which makes them resort to injection drug use. So they're already disadvantaged people, and Canada does a fine job trying to protect them from HIV and HCV and hepatitis B, and we are certainly trying to develop a vaccine to protect them from Hep C. The Nobel was awarded for the discovery of the hepatitis C virus, and that was work that I did at a US biotech company in the Bay Area of San Francisco. The company was called Chiron, and I worked on it in my lab for 7 years. And I don't want to go through all the things we did to try to identify it. I had an interesting question very recently from a student who was well-intentioned, and it's always good to be challenged by students. And she said, "Why did it take you so long?" And I said, well, really, the reasons are on this slide. Really, it was then known as non-A, non-B hepatitis. In other words, it was a common hepatitis that was not due to hepatitis A or hepatitis B, and nobody knew what it was, so they called it non-A, non-B. And obviously we had human samples, blood and liver biopsies from humans with the disease. And then various groups around the world developed a chimpanzee model. And so there were samples from that species. But still to this day, the chimpanzee is the only reliable animal model for Hep C, unfortunately. And to this day, it's still a very difficult virus to grow in cell culture. So back in 1982, students will be appalled to know that PCR had not yet been discovered, polymerase chain reaction, and that would have been very handy in the 80s to have had. It wasn't till towards the end, pretty much until we discovered the virus, that PCR started to become into routine use. There was no antigen identified for non-A non-B, as in the case of the Australia antigen, so-called discovered by Baruch Blumberg, which turned out to be the surface antigen of the hepatitis B virus. He discovered it in Australian indigenous peoples, called it the Australia antigen, but turned out to be the viral antigen itself. Nobody had an antibody defined for the virus, no cell culture system, no high throughput sequencing, and certainly no deep sequencing that is available now. So it's kind of ironic in a way. If we were trying to discover it now, it probably would take 7 weeks, but back in the 80s, it took us 7 years because there was no molecular handle available. So I don't want to bore you with what we did for 7 years. We found some interesting things, and we got some interesting publications along the way. But in terms of identifying Hep C, we did one successful experiment in 7 years, and this is it, shown here on this slide. On the top left, we were collaborating with Dr. Dan Bradley at the Centers for Disease Control. He was an expert in the chimpanzee model, and he provided us with some infectious chimpanzee plasma that could actually transmit disease to other animals. So I took that and I diluted it 6 times, and then I ultracentrifuged such that the smallest known virus would be pelleted. We didn't know what kind of virus it was, so I extracted RNA and DNA from that pellet, and then I cloned it into a lambda GT11 cloning vector developed by Drs. Young and Davis out of Stanford University. And it's not only an efficient cloning vector, but it's also an expression vector. So it expresses the cloned nucleic acid into protein. And so essentially what is produced is a large proteomics library, each expressing a protein from a piece of nucleic acid present in that pellet. And then we took serum from patients diagnosed with non-A non-B, and we presumed, hoped is a better word, that there were antibodies to the virus. And so we incubated the proteomics library. You blot it on a filter papers and so forth, and then you incubate the filter papers with the serum, the diluted serum from patients. And of course, we're looking for an interaction that we could detect using a radioactive second antibody. You do tend to get false positives using that method, and we certainly got some. But in this particular experiment, and by the way, this is the only cDNA library that I made that actually contained non-A non-B viral clones, hepatitis C clones. It was very difficult to clone the minute amounts of viral nucleic acid without PCR at that time. But my colleague, Quilim Chu, identified 511 on one of his radio autographs. And it was only a small clone, 100 base pairs. But we could show that it was derived from the genome of hepatitis C in the following way. We showed that, first of all, it was not derived from the human or chimpanzee genome. We used southern blots, and you couldn't hybridize it, whereas a single copy gene was loud and clear on the signal. We showed that the 511 clone was derived from a large RNA, around 10,000 nucleotides, which was only present in infectious samples, not in control samples. And then furthermore, we showed that that clone encodes a protein which binds antibodies found only in non-A non-B hepatitis infections, not in hepatitis A or hepatitis B. And then furthermore, as we warped the genome using hybridization methods, we could see very faint distant sequence identity with flaviviruses like dengue. And that's when we knew we had identified a virus that's causing parenteral blood-borne non-A non-B hepatitis. We then worked with groups all around the world who sent us serum panels from their patients. And Harvey Alter, who was a co-recipient of the Nobel Prize, was a leader in non-A non-B in the 70s and 80s, and he sent us some very important panels. And we also got important sera from all other parts of the world. And the conclusion was that what we had cloned was the hepatitis C virus, and it was the major cause, if not the only cause, of blood-borne non-A non-B hepatitis. And yeah, I just want to acknowledge I was very honored to get the Nobel Prize along with Harvey Alter and also Charlie Rice, who's at the Rockefeller, who's a brilliant virologist. He's really, I would say, led the field of virology in the last 25 years. But the discovery of the virus, as I've indicated to you, was a team effort. And I had two colleagues at the company I was working at, Dr. Kwee-Lim Chu, shown here, who doesn't seem to age, and Dr. George Kuo. So these were my two co-discoverers at the company I worked at. And as I mentioned, we had invaluable help from Dr. Bradley at the CDC. He provided us with a continual supply of chimpanzee materials, some of which were well characterized. So I'd like to acknowledge their role, otherwise I probably wouldn't be talking to you right now. So identifying the genome of Hep C really opened up the field of hepatitis C. It turns out that the genome's around 10,000 nucleotides. It encodes a bunch of proteins required for replication in the non-structural region. The NS proteins are shown here. And the genome also encodes three viral proteins, structural proteins, the nuclear capsid right at the end of the polyprotein, and then two envelope glycoproteins, E1 and E2. So the virus makes a large polyprotein precursor that is cleaved co- and post-translationally by host enzymes like host signalases that cleave the structural proteins from the precursor, and then two viral encoded proteases that cleave the non-structural part of the polyprotein. So the field of Hep C, I think, started with this genome identification and isolation. And the first thing that we did, we needed to prevent blood transmission. So at that time, if you had a blood transfusion, typically multiple units would be used. You normally don't get a transfusion unless you need multiple units. And the chance of walking away out of that hospital with non-A non-B was around 5%, 1 in 20. And so our first obligation was to develop blood tests to prevent that. And it turns out 511 is derived from non-structural protein 4A. It has a very immunodominant B-cell epitope in it. And when we walked along the genome, we identified additional B-cell epitopes. We also showed that the 5' untranslated region is an internal ribosome entry site, highly conserved amongst the Hep C viruses, which are very heterogeneous, being an RNA virus. But through a combination of antibody-based diagnostics using these B-cell epitopes all the way through the polyprotein, as well as the using the 5' untranslated region in PCR and TMA tests, we were able to completely stop post-transfusion hepatitis C. And I know in the USA, prior to our introduction of blood testing, there was something like 200 infections per day in the US blood banks. And it was very good that we were able to stop that. And we did that within 2 to 3 years of identifying the viral genome. And here's a slide, I think, from the CDC. It's out of date, so I apologize, but it's looking at the impact of our tests on post-transfusion hepatitis C. This is when our first test was introduced. You can see that produced a drop in incidence, and then our second test was introduced in 1992. Actually, our first test was introduced in 1990. I'm forgetting some of the history myself. And then when the second test was introduced, that dropped transmission even further. And then I think we ended up with like 5 blood tests all in all. And after the 5th, to my knowledge, there's been no report of post-transfusion hepatitis C anywhere. The genome identification obviously enabled academic investigators as well as corporate investigators allied with public health like NIH and CDC to start trying to develop therapeutics specific to the life cycle of the virus. And the first group to do this was Gilead. Around 2015, they came up with 2 drugs whose name is obscured on my title, but they target the RNA-dependent RNA polymerase of the virus, as well as NS5A, encoded by the virus. So a combination of these 2 specific antivirals was able to actually cure nearly 100% of patients. This was reported by our Canadian colleague Jordan Feld and colleagues in New England Journal of Medicine, as well as Graham Foster at the same time. And importantly, not only was this drug combination able to cure people, it could actually cure all the major genotypes of Hep C that occur around the world. And there's only a slight problem with genotype 3, which, as you can see, 95% of people were cured, not 100 for genotypes 2 and 3. But there are now different combinations of antivirals targeting the viral life cycle. And if rarely a patient is not cured by one group of antivirals, they're put on another cocktail, and normally that cures them. We have drugs targeting the protease, the serine protease, the chymotrypsin-like protease of the virus. And so we've effectively, when I say we, I mean the whole group of academics and private sector and public health and national institutions like NIH who have supported this work. We can now cure nearly all patients. Excuse me. That raises a question. It did back in 2015 when this great antiviral work was published, but still people ask this question: why do we need a vaccine when we have curative drugs? And it's a good question. And I think the history of infectious disease has taught us that a vaccine is definitely needed if you want to stand a chance of eliminating an epidemic. Today, the only infectious diseases that have been eliminated from the planet are smallpox and bovine rinderpest. Many other diseases have been severely curtailed and very effectively controlled, but to eliminate, you do need a vaccine. I think that's one of the lessons of infectious disease for us. And also, there are examples where even if you have effective therapeutics to infectious disease, and even if they're cheap, you know, just a few dollars, you still get outbreaks, serious outbreaks of infectious disease. And I'm grateful to Dr. Stephen Shafran, who's an MD in our university, who provided me the following few slides. What they show is the, and unfortunately I can't see the titles, but these slides are looking at the incidence of chlamydia, gonorrhea, and syphilis over time. And you can see whichever one this is. Hopefully you can see it. Can you see it, Dominic? Can you see the title of this slide?
Yes, it is reported overall and sex-specific rates of infectious syphilis from 1993 to 2011 in Canada.
Okay, I just wanted to make sure that the audience was seeing the title. That's good.
Yeah.
Yeah, you can see that the incidence of this disease increased dramatically in 2002, and increased in prevalence more than tenfold. Now, this is an infectious disease where for a few dollars we can cure the symptoms and the infection, the disease. but we don't have a vaccine. And the same applies to this disease. And as you can see, from 1997, there's been a large increase, a twofold increase in incidence of disease. Once again, we have antibiotics to cure this, but it does not prevent outbreaks, obviously, because it's not a vaccine. And also with this bacterial, with this disease, this sexually transmissible disease, once again, you see a big increase since 1997. So these are just examples where clearly cheap therapeutics are very helpful and useful, but they're not preventing, obviously, disease. So to me, it's an argument we do need a vaccine. There are 2 to 3 million infections around the globe of Hep C, and I don't think it's feasible. I hope it's possible, but I don't think it's feasible we can really curtail that kind of transmission frequency without a vaccine. Now, Hep C is an RNA virus, so it replicates through an RNA-dependent RNA polymerase, and that does not have proofreading ability like DNA viruses and like our own DNA polymerase is replicating our genomes. So you get a lot of mistakes, and the virus likes that because then it can adapt better to different situations. That's a feature of RNA viruses. They're highly mutable, and you start seeing different clades and subclades around the world. This slide from Dr. Fang, published some years ago, I'm using just to illustrate, in North America, the most common genotype is genotype 1, and the most common subtype is 1A. It accounts for maybe 60 to 70% of infections in Canada and in the United States, followed by 1B, oh dear, 1B and genotypes 2 and 3. So around 70% are due to genotypes 1. If you go to North Africa, for example, Egypt, genotype 4 is predominant. And unfortunately, that is somewhat of a tragedy itself, because the Egyptian population were treated for schistosomiasis, sleeping sickness, many decades ago. And so they were treated with, I can't recall actually if it was a therapeutic or a vaccine, but unfortunately the way it was given to the population was not done aseptically. And what resulted was a huge epidemic of hepatitis C in the country. Something like 15 to 20% of Egyptians are carrying the hepatitis C virus, and it's clade 4. So at the nucleotide level, these clades vary by up to 30% sequence diversity. So they're really quite different, but of course they're still Hep C variants. If you go to South Africa, there's a genotype 5 that predominates. And if you go into Vietnam, genotype 6 is very common. The most common genotypes are 1A and 1B around the world. But in developing a vaccine, we have to take into account the diversity of the viral species, the viral genus, I should say. So for the last 10 years at the University of Alberta, I've continued my work on trying to develop a vaccine against Hep C virus. I spent a lot of time in the US working on it. And in the US, we ran 4 clinical trials of different vaccine formulations with different adjuvants. The one that's really looking the most promising, and I think is very promising, is one that contains recombinant envelope glycoproteins, GPE1 and GPE2. There are 2 envelope glycoproteins encoded by the virus. They're highly glycosylated, but we're able to make them in transfected mammalian cells. They associate together in the lumen of the ER. They are not secreted in our transfected cells, but of course in the virus-infected cells, they do escape from the ER by budding out of the ER and then secreted into the blood. So the envelope glycoproteins, we make them in Chinese hamster ovary cell lines, and we've tested them. When I was in the States, we tested them in the chimpanzee model, and those antigens remain to this day the only vaccine candidate that is shown to be protective in the chimpanzee model, or for that matter, in any other model. There are really a dearth of models for Hep C, as I mentioned. So nobody has shown any other vaccine to be protective in the chimpanzee model. And what the vaccine does is it doesn't necessarily sterilize from infection. Many vaccines do not do that. I think it's a little bit of a mythology that vaccines sterilize. They usually don't. What they do is stimulate an anamnestic immune response to eradicate the viral infection, which may be subclinical and a very low level. And that's what our vaccine does against Hep C. It can sterilize, but often it just curtails acute infection without proceeding to persistence. It is the persistence of Hep C over many years that is associated with the pathology, chronic hepatitis, liver cirrhosis, and in some cases, liver cancer. So what we want to prevent by vaccination is the persistence of viremia. And I think we can do this with this vaccine. This is just a gel of E2. You can see E2. It's highly glycosylated, and it has a complex carbohydrate composition, which accounts for this smear on jelly electrophoresis, and this is E1 here. And as I mentioned, they associate together to form a heterodimer. This is work done in the US in collaboration with the NIH, and it summarizes all of our data using E1, E2 made in mammalian cells as a vaccine antigen, to which we add a adjuvant. Usually it was an oil water squalene-based emulsion. Sometimes we used some immunostimulants like MPL, monophospholipid A, which stimulates Toll-4 ligand. So it's a little bit of a potpourri of all of our studies in chimps. You obviously don't want to do too many studies in chimps for various reasons. But what they showed is when we vaccinated and then we challenged a few weeks later with homologous virus, shown here, we had 12 vaccinees. 5 of them were sterilized from infection, and those were the ones that made the highest antibody titers to our vaccine. It was very clear. But of the remaining 7 that became acutely infected, only 2 went on to become chronically infected. And so only 17% became persistent carriers of the virus. In unimmunized controls, they all got infected, 10 out of 10, and 70% developed chronic persistent infection, which is in humans where the pathology comes in. And you can see using Fisher's exact test, this was highly significant. Well, very significant, perhaps not highly, but 0.03. And then when we challenged with heterologous 1A virus, and I would emphasize 1A is the most common genotype in Canada and USA, of 19 vaccinees, we didn't sterilize any of those, unlike with the homologous virus, but only 3 went on to develop chronic persistent infection, 16%. Of 14 controls that were unimmunized, 57% went on to develop chronic persistent infection. So once again, a significant difference statistically. So we want to ask the next question, which is, does this vaccine elicit broadly cross-neutralizing antibodies to the different clades seen around the world? We sent our chimpanzee antisera to the NIH, and J.C. Mounier in Bob Purcell's lab looked at this question. We sent him 4 chimpanzee sera, and the different colors represent different genotypes of the virus that he tried to neutralize. So the antisera from the chimps was diluted 50-fold. And then he looked at the percent neutralization. And you can see that genotypes 1, 4, 5, and 6 were highly neutralized by the antisera. Genotype 1 is the most common genotype in the world. Genotype 4 occurs predominantly in North Africa, genotype 5 predominantly in South Africa, and genotype 6 predominantly in Vietnam. Genotypes 2 and 3 that account for maybe 20–30% of infections in the West were neutralized, and you're looking at groups of 4 different chimpanzee antisera here. That's why you have 4 bars. You can see that genotypes 2 and 3 were not as well neutralized. They were partially neutralized, but not as well as genotypes 1, 4, 5, and 6. But I think the important thing here is that a vaccine derived from a single strain can actually neutralize multiple clades. And so, you know, this was an important breakthrough, I think, because a lot of people, including NIH, CDC, were very concerned about the heterogeneity, not being able to produce a global vaccine. Now, at the University of Alberta, my excellent colleague, John Law, who's a very fine virologist, he looked at clinical samples. These were humans who were vaccinated with E1, E2 in the United States. And John addressed the question, do those people make cross-neutralizing antibodies like the chimpanzees did? And we published in 2013, we looked at 3 individuals that received the vaccine, individuals 1, 5, and 7. The different colors represent the different genotypes seen around the world. And if you look at individual 5, you can see clearly that we were able to neutralize genotypes 1A, 1B, 2A, 2B, 4, 5, and 6 very well. A little deficient in genotype 3, but at least it was partially neutralizable. And we could see the same pattern in individual number 7, and not as well to individual number 1, but it actually kind of confirms that genotypes 2 and 3 are not as well neutralized by our vaccine as the other clades. Running a little short of time, so I'm going to go quickly over these slides. Basically, what we've shown in the last few years is that the reason we get cross-neutralizing antibodies is, as you would expect, as you would surmise, there are highly conserved neutralizing epitopes in E1, E2. And we've shown in animals that we vaccinated and in humans that indeed all species make antibodies to those highly conserved cross-neutralizing epitopes. And that is why we can see such a broad cross-neutralizing profile. And I don't have time to go into that. I just do want to mention in the context of a Hep C vaccine that cellular immunity is also important. We know that in protection from studies performed back in 2003. So we are dialing in some T-cell responses to highly conserved epitopes encoded by the virus. And so we're trying to focus the immune response on highly conserved T-cell epitopes. And so we're providing synthetic peptides to those highly conserved CD4 and CD8 epitopes along with E1, E2. And in this way, if you use a collection of synthetic peptides, you can actually cover more than 90% of the population with respect to MHC class 1 diversity. So we should be able to elicit cross-neutralizing antibodies and cross-reactive CD4s and CD8 T-cell responses in most of the population. We're using a suitable adjuvant, of course. So this vaccine now is being put together in the ACTM at the U of A, the Alberta Cell Therapy Manufacturing Unit, run by Greg Corbett. We're hopefully about to finish making the E1, E2 expressing cell batches under GMP. And we'll be putting the vaccine together with the peptides and the adjuvant probably in spring, early summer. We obviously, like most of you, we have a halt in our work right now because of COVID. But we've got a lot of partners willing to test our vaccine in the clinic, the US NIH, the Helmholtz Institute in Germany, as well as University of Pisa in Italy, and an Australian center also. So we'll be doing, hopefully, phase 1 at the end of next year. Now, turning to COVID, I think most of you, I'm sure, know what's happening with COVID vaccination, but there's a few points I did want to bring out in this talk. And as you know, new technologies are one of the reasons why COVID vaccines have been developed so rapidly. The virus was only discovered by deep sequencing back in January of this year, and already in December, Canada received its first vaccine approved by Health Canada. That was on Sunday night. And there are two basically new technologies being applied to COVID. The one from Oxford University using defective adenoviruses to deliver the spike protein, and they're working with J&J, they're working with AstraZeneca. J&J and CanSino also have similar types of vaccine delivery systems that they're applying. And then, enter the RNA vaccine technology from Moderna and BioNTech and Pfizer. This is remarkable because this is a really new technology. It really has developed only in the last 2 to 3 years. There's no approved vaccine other than COVID. So it's an amazing example of how technology can really enable progress. So the trick with the RNA vaccines are several. First of all, Moderna and BioNTech, they have to modify the nucleosides in the RNA so as to reduce the innate immune response. If they don't do that, then expression from the RNA is highly reduced, and you don't get a good immune response. So they have to incorporate modified nucleosides to prevent a strong innate immune response. They have to add a cap structure to the 5' end of the RNA to increase translation efficiency. And then they have to protect the RNA by making a rather complex lipid formulation, which not only stabilizes the RNA, also makes a viral-like particle of about 120, 140 nanometers, which is good because the immune system recognizes it as a viral particle, and that's what you want. There are other approaches that are ongoing. Novavax are using recombinant spike antigens with adjuvant. Medicargo producing the spike protein in plants, and recently got a large grant from the Canadian government to produce their vaccine. And Sanofi GSK also have recombinant spike antigens with adjuvants as their vaccine. And there is also some work being done with DNA vaccines by Inovio. Also, John Lewis at the U of A is trying to develop a DNA vaccine for COVID. So this is work from, I think, Dr. Wall's group, published earlier this year. And it's showing what happens when the spike protein is made in infected cells. You actually form a trimer. So this is a trimeric complex. So it's a very high molecular weight complex. And the part of the spike that interacts with the receptor, angiotensin converting enzyme 2, is shown here in green. What's interesting is only one of the receptor binding domains is accessible in the trimer. The other two, shown, I believe, here in blue and red, they're actually not accessible. So it's an interesting molecular architecture that only one of the receptor binding domains is available. And here's a schematic of the spike protein. It gets cleaved in the ER by furin to produce the S1 subunit and the S2 subunit. The receptor binding domain is in the S1 subunit, and it forms this nice trimer. So Moderna express they express the full-length spike protein in their RNA vaccine, and 1 or 2 months ago they reported in phase 3 trials it was 94% effective at preventing COVID disease. And then BioNTech and Pfizer followed up with their phase 3 trial results, a similar kind of vaccine, different lipids, but once again, an RNA expressing the whole spike protein. They found very similar efficacies, 94%, in preventing infection following a 2-dose vaccine schedule. Now, the efficacy so far that's been reported is the efficacy seen within 1 to 2 months after the 2-dose vaccination schedule. It remains to be seen how good that's going to be in the future after 6 months and 12 months and so forth. So this is an early read on efficacy, but it's very encouraging to see 94%, albeit after just 1 or 2 months of vaccination. And then also BioNTech and Pfizer reported that it was 94% effective in people over 65 years of age. So that's very important, because as you age, immune responses diminish. It is well tolerated. You typically get vaccine side effects that you get with many other vaccines against different pathogens. You get some injection site pain, you get a little bit of fever and a little bit of headache. I had the shingles vaccine a few weeks ago, and that's exactly what I experienced for a few days. So these side effects are very common to many other vaccines against different pathogens. And on December 9th, the Pfizer vaccine was approved for human use in Canada, emergency use in Canada, and it was delivered on 13th of December, which was last Sunday. And people, the healthcare workers, the first responders are now being vaccinated this week in Canada. So great potential for RNA vaccine technology. It can be applied pretty much to any vaccine against any pathogen, except bacterial vaccines where you're targeting antibodies to the capsule, the polysaccharide capsule. But they have a wide application, and I think it will transform the vaccine industry in the future and will be a way to make vaccines efficient and also any emerging disease, as we've seen with COVID, can be vaccinated very quickly using this technology. But I think there are some important questions that we need to keep our eye on. And obviously the first one is duration of immunity. How long will the immunity last? I think if you look at influenza vaccines, you do see that eventually antibodies do decline. That's very normal for the immune response. You don't expect immune responses to stay up, elevated all the time. They will decline, and it wouldn't surprise me if we will need annual boosters of the COVID vaccine, which is not necessarily a problem, but I think it's likely we will do that, excuse me. And also, I think if RNA vaccinology is rolled out to many different vaccines, we will have to address the issue, well, it's safe for COVID, but if you start doing it 10 or 20 times for different vaccines, is it still safe? And so that will be an issue. I'm not saying it's going to be a problem, but I'm saying it has to be obviously addressed. And then we still actually don't know which is the most potent vaccine against COVID. Is it RNA? Is it adeno? Or is it adjuvanted recombinant proteins? We actually don't know which is the best at eliciting antibodies and T cells. And part of that, when you're talking about vaccinating the world, and perhaps with annual boosters, cost of goods becomes an essential factor. And we need to figure out, which is the most economical to do this. And finally, but not least of all, there's the issue of stability of these vaccines. The Pfizer BioNTech vaccine has to be shipped in dry ice at -70. The Moderna vaccine, RNA vaccine, can be shipped at -20, which is better. But when they're thawed at 4 degrees, the Pfizer vaccine, I believe, is stable for a few weeks. And the Moderna vaccine is more stable when it's thawed at 4 degrees, probably for 2 or 3 months or so. I don't know exactly, but I know it's more stable at 4 degrees than the BioNTech vaccine. If you compare it to adeno-based vaccines and recombinant protein vaccines, those can be stored at 4 degrees. They're very stable. So in terms of getting these vaccines out to all the countries of the world, many of which have poor infrastructures and weak economies. It may be the adenovector vaccines and the recombinant protein vaccines that do need to be used in those countries. What we've done at the U of A in the Li Ka-shing Applied Virology Institute is we focused on the receptor binding domain as a vaccine. We make it in Chinese hamster ovary cell lines. We could express it at very high levels relative to the whole spike protein. And so, you know, when you're looking at the challenge of providing vaccine to everyone in Canada, 33 million people, the expression level is very important. So we decided to go for the receptor binding domain, and we've immunized animals. This is, you're looking at neutralizing antibody assays performed by John Law, and he worked with Mike Logan and other colleagues to do this work, Darren Hockman as well. We're looking at what happens when you immunize mice with the receptor binding domain made in CHO cells. We added an adjuvant, alum MPL, as well as an adjuvant called ASO3. And you can see that when we dilute the antisera 450 times, we get zero infection of COVID pseudoparticles. We completely prevent the entry of COVID pseudoparticles expressing the spike protein into receptor cells. They just don't get in because of neutralizing antibodies. And even at dilutions of 1350, we start to see a little bit of entry, but we get more than 95% inhibition of cell entry by the pseudoviruses. And it's not until you get a dilution of 1 in 36,000 that you start to see only 50% inhibition of cell entry. So this little protein with a nice adjuvant, and these adjuvants have been approved for human use, so they're safe. This is a good candidate for a COVID vaccine. I should add, Lorne Terrell's lab has shown that also our antisera can neutralize the infectivity of the actual virus in cell cultures. So we've confirmed that these are true high neutralizing antibody titers. Now, a Chinese group published recently that a very similar vaccine that they're working on, the receptor binding domain, they also agree with us that it's a good vaccine for COVID, and they've actually gone and shown it is efficacious in animal models that they vaccinated and then challenged with the virus. So I think this is a good COVID vaccine candidate, and potentially we could manufacture this for clinical use in certain countries, ones that I think need a stable vaccine that can be shipped at 4 degrees, and countries where perhaps they don't have the infrastructure to make their own vaccine. We certainly would be happy to work with any countries. We could provide our cell lines, and we could manufacture our vaccine for certain countries and provide it to them. So if there's any interactions that you have with countries like that, do let me know. I think with Canada, we're going to go ahead and make some under GMP because for 2 reasons. One is we still don't know which is the best vaccine. It could be this one. We don't know. And secondly, probably we are going to have to boost Canadians on a regular basis. That's my prediction. It may not be true, but I think it's likely we'll need to boost people. And so we're going to need a lot of vaccine, and cost of goods becomes an important issue. I'm nearly finished, so thank you for your patience. I want to just finish with this question. Would a vaccine stockpiled against the SARS coronavirus 1 that emerged in 2003, if we had made that and stockpiled it, would it have prevented the COVID-19 epidemic? The answer is yes, it would have done. And this is work from Dr. Walls, who published a very nice paper in Cell this year. What he did is he immunized mice with recombinant spike protein from the 2003 coronavirus. He used an adjuvant, I can't remember which one now, and then he assayed for the ability of the antisera to neutralize the infectivity of that 2003 strain. So he's presenting data here on 4 mice, 1, 2, 3, 4, and compared with control, where COVID pseudo particles could enter the cell very easily. None of these mice allowed that. Their antisera completely neutralized the infectivity of these pseudoparticles. Then he said, well, can the antisera neutralize the infectivity of COVID-19 pseudoparticles? And here's the control where entry was very efficient. And in the 4 mice, you can see that the antisera didn't completely prevent entry of the cell, into the cell by the COVID pseudoparticles, but it was 90% inhibited. And so I think it's very likely that if we had made the SARS 2003 vaccine and kept it stored, they are very stable, recombinant proteins. You can keep them at -80 forever, pretty much. If we had that available, we could have prevented a lot of the disease and a lot of the fatality of the COVID epidemic. And so that's a very important lesson we have to learn. When these strains emerge, don't stop manufacturing the vaccine because the epidemic has gone away, which what happened with SARS 2003. Make the vaccine because there's likely to be another coronavirus that enters the cells in a similar way, and you can get at least partial protection with the earlier vaccine. How should we prepare for the next epidemic? I think I've already dealt with one proposal. Stockpile vaccines as these viruses emerge. Don't forget about them when they go away. I think COVID has indicated to all of us that we need more funding for basic research in infectious disease and in translational research in infectious disease. We are under constant threat, and all virologists agree that there will be other pandemics that affect humans. We ought to fund more infectious disease research. I think, as many of you know, we need to increase Canada's manufacturing facilities. We don't, at the moment, have a great resource of manufacturing capabilities. And so that makes us dependent on other countries. We need to become more independent. We need to be able to manufacture RNA vaccines as well as recombinant protein vaccines. And I think we need national labs to do that. And finally, we all need to come together. We cannot be accusing people of causing epidemics. We all need to get together in an international forum and discuss what public health measures can we enact to minimize the risk of future epidemics. As many of you know, a lot of the pandemic flu strains, the reservoir for those strains are in aquatic birds. The coronaviruses replicate in bats and depending on the strain, civet cats and so forth. But part of the problem is there's a lot of human interaction with those reservoirs. And I think if we got together and discussed sensibly and maturely, we can probably enact some public health measures to reduce the incidence of future pandemics. And I think these are things that we need to do now in preparation for the next pandemic. And in closing, I'd like to acknowledge many people in my CERC lab and who are now in the Li Ka-shing Applied Virology Institute. John Law is an excellent virologist that's done a lot of really good work on Hep C and COVID. Mike Logan is a great cell biologist, a molecular biologist that's worked on both programs. Amir Landi is a great cellular immunologist, and I don't have time to go through the whole list, but teamwork, as always, is essential. And we've got a fine group of young scientists in the Li Ka-shing Applied Virology Institute, many of whom used to be in my Canada Excellence Research Chair lab. And this is a group that's going to be very helpful to stem future pandemics. And finally, I'd like to thank Lorne Tyrell, who I've worked with closely ever since I've been in the University of Alberta for the last 10 years. And he's a great colleague and a great mentor and friend. Thank you very much. I'm tested your patience. Sorry I went on so long.